Good afternoon, everyone. It is my pleasure to welcome you both to the last seminar we have for the semester and to the 2024 Warren K. Lewis Lecture in Chemical Engineering here at MIT. Before I introduce our honored lecturer for this afternoon, I want to say just a few words about the person for whom this lectureship is named. Doc Lewis, as he was affectionately known by his students and associates, had an enormous impact on shaping this program and the field of chemical engineering as a whole. Doc entered MIT in 1901, where he first met William H. Walker, head of the Course 10 program in chemical engineering that then resided within the Department of Chemistry. He received his PhD in physical chemistry from the University of Breslau, Germany in 1908, and joined MIT as an assistant professor in 1910. He was promoted to professor in 1914. Doc Lewis was the first head of the newly formed independent chemical engineering department from 1920 to 1929. Lewis revolutionized the design of chemical engineering equipment with the concept of unit operations, the first paradigm of chemical engineering. He was an eminent researcher and inventor. He contributed much to the field of industrial chemistry with over 80 patents to his credit. As one example, he pioneered the use of fluidized bed catalytic cracking and refining. Above all, Doc Lewis was a superb educator. His text, Principles of Chemical Engineering with Walker and McAdams in 1923, first defined the discipline. His lectures are legendary for the combination of beautifully organized material and Socratic exchanges with his students. And actually, a fact that I found out just a few months ago, Doc Lewis was recognized in a series called Great Teachers, featured in a special issue of Life magazine, published on October 16, 1950. He was one of 8 professors, 7 of whom were men, that were featured in this issue, and those that were honored were chosen by then current college students. So I think it's instructive to think that even back in 1950, his impact on the students here at MIT was being felt substantially enough to be recognized in a national magazine. Not surprisingly, Lewis won many awards, among them the President's Medal of Science, the President's Medal of Merit, and the Priestley Medal from the American Chemical Society. As a most fitting honor, our professional society, AICHE, has honored him with the creation of the Warren K. Lewis Award, which recognizes outstanding education in chemical engineering. Following his term as department head, he devoted himself to teaching, research, and consulting, and remained an influential member of the department until his death in 1975 at the age of 92. The Warren K. Lewis Lectureship was established here at MIT in 1978 to recognize Professor Lewis's revolutionary impact on chemical engineering. By developing the concept of unit operations, first proposed by A.D. Little and William Walker, he revolutionized the design of chemical engineering processes and equipment. Throughout his career, Professor Lewis was mindful of the needs of industrial practice. Accordingly, the Lewis Lecture features speakers from both industry and academia. And now, it is my pleasure to introduce this year's lecturer, Dr. Jean Tom. Jean is currently the 2024 Brenton Halsey Distinguished Visiting Professor of Chemical Engineering at the University of Virginia. She received, I wrote down BS degrees, but they are actually SB degrees in chemistry and chemical engineering from here at MIT, and an SM degree in chemical engineering practice from MIT. So for those of you who are practice school alums in the department, you can certainly have a conversation with Dr. Tom about her experiences. After several years in industry, she returned to academia and received a PhD in chemical engineering from Princeton University. Jean spent 19 years at Merck Research Labs before joining Bristol Myers Squibb in 2006. At BMS, she was the executive director of development engineering, chemistry process development, leading a group of chemical engineers focused on development of chemical processes to synthesize small molecule drug candidates for new therapeutics. Jean recently retired from BMS prior to starting her current position at UVA. Now I have pictures Jean doesn't know I have. Jean is very active in AICHE. She's a board of directors member, she's a fellow, and she's a recipient of the AICHE Industrial Leadership Award given in 2018. She's also active in the National Academy of Engineering, to which she was elected in 2019. She contributes to the chemical engineering community through her service as an ABET program evaluator, external advisory committee for several departments, and through STEM outreach programs. And you can see in the pictures that we have here, there's her with Paula at the AICHE Doing a World of Good medal ceremony. This is her recently at an event in Charlottesville for students in grades 4 through 8 called Girls Excelling at Math. And there she is with the Cavalier. I think I'm supposed to say, is it Wahoo Wah? There you go. I'm supposed to say Wahoo Wah. Is that right? I never know if it's that or Who Wahoo, but Wahoo Wah at UVA. Jean has been no. Jean has been a very strong advocate for increasing the role and impact of chemical engineering on the pharmaceutical industry, bringing new approaches to accelerate the generation of process knowledge needed at the different stages of development, and increasing the role of pre-competitive collaboration to advance new technologies for the development of new compounds. And I have to add a personal note here, which is that I first met Jean a few years ago as we were both on this National Academy's Consensus Study report. And I saw firsthand through our work on this report how she advocated not only for chemistry and chemical engineering, you can see the central themes there, but especially for that last one, preparing and empowering the next generation of chemical workforce. She has been a tireless advocate for advancing opportunity for talented individuals. She and I actually together spoke at an AICHE meeting about this report and its focus on workforce development, as well as at an ACS diversity seminar. And I'm sure through her presentation today, you will learn other strategies, especially for those of you who are students and postdocs in the room, about how to advance your careers in a very inclusive way. Please join me in recognizing and welcoming our Warren K. Lewis lecturer, Dr. Jean Tom.
Okay, is this on? Yes. All right, thank you, Chris, for that really kind introduction. I am delighted and thrilled to be here at my alma mater, and the last time I spoke in this room was in 1984 when I was giving my project presentation. I think it was 1026 then, and I was really nervous then, a little less nervous now. This is the Warren K. Lewis lecturer. He is known as the father of chemical engineering, America's first chemical engineer. And I thought, well, he's the first chemical engineer. Is there a possibility I'm related to him? And so I went and searched the lineage, and Tom Sherwood, another MIT professor, was a student of his, and then Glenn Williams, and then Charles Sattersfield, Bob Reed, Pablo De Benedetti, and I am a student of Pablo De Benedetti. So I'm a sixth-generation descendant of Warren K. Lewis, which I did not know before being asked to give this lecture. Very proud to be here. So I was a freshman here. I don't know if they still take the freshman picture, but that's what it was. And this is what course 10 looked like for me. 111 students, of which 29% were women. And I went back and thought, oh my gosh, I only can think of 2 women professors I had, and they were both in the chemistry department. I did live in McCormick, so that was a really great support network for me as a woman student here. And my fellow class of '84 and McCormick resident, you know well. I then did spend a year getting the practice school degree. It was in Bethlehem Steel and General Electric. Both sites I don't think are Bethlehem Steel or General Electrics anymore. But this was really a transformational experience for me to really understand what chemical engineers can do and how to solve these open problems. And I'm still in touch with the practice school director and assistant director who was at GE, and invited him down to come lecture in my class. So just a little preamble before the talk. So the talk today is about the development of small molecules being discovered for therapeutics. And what I wanted to do was give some examples of innovation as we develop the process that's going to be the manufacturing process. And we go through what I describe as these steps here. And there is an aspect of data science and modeling that is becoming more prevalent in the work that we do. I want to talk about collaboration, how that's driving innovation, but just as important as collaboration and the new tools that we're using is really thinking about how we develop people, the culture in which we do this work, and engaging the next generation to enable that innovation. So drug substance, I think everyone probably knows this, that there's really what is considered two types of molecules: small molecules, which are made synthetically, and large molecule, which comes through biologic routes. So small molecule through a series of chemical reactions, biologics through the host cell. And so the molecular weight differences that you see here. And biologics get a lot of press. You hear about them a lot. But what is actually being approved is a lot of molecules and a lot of small molecules. And so this is the data from what's been approved from the FDA over the past 30 years. And I took a look at what was approved in the first 10 years of this time period and the average number of approval, the distribution of biologics versus small molecules. It's about 86% small molecules, 14% biologics. And then looking at the last 5 years, you can see this tremendous productivity in the pharmaceutical industry, a doubling of the number of biologic as a proportion of these molecules. But because the productivity has increased so much, we're actually seeing about 60% more small molecules. And what you don't see in this is sort of the time dependence, is that the time in which we can develop these molecules have shrunk. So the number of molecules that we're working on at a given time is increased even further. And so what do some of these small molecules look like? Well, they are very complex to make synthetically. And these are some of the molecules that have been approved in the last few years. The one in blue are ones from BMS that my group had a opportunity to work on. And what you see is the complexity in the form of a lot of heterocyclo macrocyclic molecules, molecules with 3D dimensionality to it, molecules other than carbon, nitrogen, oxygen, and hydrogen. So there's sulfur incorporated, there's fluorine incorporated, and there's deuterium incorporated in some of these molecules. Also in the small molecule world is the ability to make these much larger molecules, oligonucleotides, peptides, and then conjugating both small molecules and peptides to antibodies. And last year, there were 9 drugs approved by the FDA, which were oligonucleotides and peptides. So that represents 16% of the number of molecules that were approved. So this is an area of tremendous growth when we think about small molecules. So how do we get to these molecules? Certainly advances in the understanding of human disease enables us to discover these new molecules. And the discovery is based on many scientific fields exploring the cellular mechanism of diseases and functions. And that really leads to the medchem folks, the medicinal chemists, who are then trying to design the most appropriate molecule to have that functionality. And that's how we get these new drug candidates. So I'm not going to talk about this discovery or medchem, but what we do once we have a new drug candidate. And that is the world of what we call pharmaceutical development, where there is a new drug, new chemical entity which has some therapeutic purpose. And what we need to do as in a pharmaceutical development organization, in which the chemical engineers are a big part of, is to develop the chemical process by which we're going to be able to make this active ingredient, this drug substance. The work doesn't stop there. We have to then come up with a process in which we can formulate this to make the drug product, which is what you take as a patient. And there's also engineering and thinking about how are we going to package this to understand the stability of the molecule. So all of this involves engineering. And the world that I've lived in for the past 35 years or so is the world of developing the chemical process, which essentially is turning raw material into these active ingredients. And so this development world is really sitting at the interface between discovery and manufacturing. And one of the great things about being an engineer in this area is the ability to work across scales and facilities, is that we will start working on this when it's coming out of discovery, and we have to change the synthesis, and we have to change the scale in which we are able to produce it, and we change the facility in which we're doing this work. And so this leads to the thought of, as chemical engineers, how do we figure out what is scale dependent and what is not scale dependent? We also work across the unit operation. So for a given transformation, we have to figure out what are the key unit operations that we're going to be using, what's the sequence in which that is happening, and understanding the key engineering aspects of it, whether it's safety, the equipment parameters, the process parameters, the material property of your material going in and going out, but also the thermodynamics, kinetics, and the heat and mass transfer. And this is very much what Doc Lewis wrote in the textbook that was sort of seminal for this field. And a lot of the topics that he talked about are still very relevant. And I would also point out, looking at this index, is that he talks about some unit operation that really doesn't get much press or oversight these days, but is really important in the work that we do: the filtration, the drying, and the what he calls crushing and grinding. Is that we actually mill the material, so that unit operation is actually fairly important. And Doc Lewis also had another book called Industrial Stoichiometry, and this is about understanding where every molecule is going in the process. So he says, use stoichiometry in the diagnosis of performance and the solution of the ills of the process operation. And that's what we do in the field, is that we have to understand what all the reaction is making and how do we get it out of the system if there are impurities. So that's very much relevant. So, it was fun to go look up these books from Doc Lewis. And so the unit operation in chemistry comes into play in a chemical process development organization. And what we're trying to do is design these safe, sustainable, cost-effective routes and processes to deliver high-quality drug substance to the patient. And we start our work coming out of discovery, and the route that we make the molecule from discovery is not something that we can scale up, because it's a flexible route that's designed to make very small quantities of material with very different, slightly different nuances to it. And so when a molecule comes out of discovery and we're thinking about phase 1, we have to think about how are we going to invent a route that could eventually be scalable. And route invention is determining what's the intermediates that we're going to use to build this molecule. If it succeeds in phase 1 and it's entering the next stage of development, we think about what we call process invention, which is finalizing the reagents, solvents, and unit operations to make that transformation. And hopefully the compound goes on to what we call late stage, or phase 3, the very large clinical trials, in which case there's a high probability it's going to be, a package is going to be put together for the FDA. And that is what we call process characterization, where we optimize the process conditions and come up with control strategies to ensure robustness. And after that, hopefully we transfer it to our manufacturing colleagues and it becomes a drug. So the way I talk about this is that, route invention is about what is the path in which we make the molecule. Process invention is what's the process, and process characterization is about the data that we need so that we can file this drug. And it's an interplay between our chemistry colleagues with engineering, having some touch points in the route invention, being a real partner with our chemistry colleagues in process invention, but in process characterization, that's usually something that the chemical engineers are doing. And so what I'd like to do is sort of give a little story about each of these aspects of the 3 different parts of process development. So the path, this is determining the intermediates to make the molecule. And when you're trying to design the route to make the molecule, there's just a lot of potential synthetic pathways. So the question is, how do we best select the option? How do we best decide what should we pursue to develop? We want to minimize the resources that we use, because we can't have every chemist working on all the different options. And we wanted to do this fairly quickly so that we don't waste time going down a path that doesn't look like it's going to be viable later on. And so, and there's a lot of optimization that we're thinking about in different criteria. And this is a really nice diagram from Connor Coley and Klaus Jensen in the paper, where we're thinking about safety, we're thinking about the cost of goods, sustainability, what unit operations, what's the supply chain for the raw material look like, and whether we have freedom to operate from a intellectual property. So really, we think of route invention as a problem in problem solving, in decision making. And one of the things that have done at Bristol Myers Squibb is to really make an expansive ideation instead of assigning it to one or two chemists to go work on this particular compound. They open it up to the entire process chemistry group and ask people to submit ideas of how might you want to build this molecule. And then they get a group together and look at all those ideas, and you start to see strategies that are similar and aggregate and decide, these are likely the strategies given that you've got a large cohort who've come up with ideas. But then with all these strategies, how do you decide which strategy to select to work on? And so one of the things that we do is think about, from a sustainability, process mass intensity, which is the total mass of material divided by the mass of isolated products. So the lower the number, the better it is from a sustainability perspective. And we can use prior data to inform us and use that to predict what might be a good strategy. And so there's been a database collected of the process mass intensity of over 1,000 reactions that have been implemented. So these are large-scale implementation, not just lab scale, as well as additional data from the literature, all that have been made at greater than 1 kilo. So it represents a larger scale. And so we can segment that data to understand for amidation or cyclization, here's the distribution of what a PMI looks like that's likely to be. And then we can look at all the different routes being proposed because they're proposing different reactions, and use that database to figure out what might be the likely PMI. And so you could sample the PMI and yield based on the correlated normal distribution assumption and calculate a PMI. And you could do this thousands of times. And then you could get a prediction of what the likely PMI is for a particular step or for a particular route. And so that allows us to then look at the different ideas and say which one is likely to have a lower PMI. And so this is a tool that actually is an app that was put together by a lot of pharmaceutical companies through the ACS Green Chemistry Pharmaceutical Roundtable. So that's just an input that enables us to think about all the different ideas in terms of route invention. Okay, so we have a route. We know what the transformation we want to do. How do we develop the process? What's the best unit operation or reagents for each synthetic step? So in this phase of development, we want to determine the details about each of the synthetic steps. And so in this palladium catalyzed coupling, your reaction space is, you can think about, you're going to need to select a ligand, you're going to need to select a base, what's the solvent, what's the temperature, what's the reaction time. So if you think of all the possible combination, and what we're optimizing against is yield, selectivity, and pure level. So if you think of all the combination, that's 10 to the 5th configuration. So you don't want to do 10 to the 5th experiments to figure out what's the right set of conditions. What we can do is we can conduct mechanistic studies, we can conduct DOE, we certainly will be doing high throughput experimentation, we could do response service modeling. But a couple years ago, in a collaboration with the Princeton Catalyst Institute, Abby Doyle and Ryan Adam, professors there, came up with a way to use experimental design by Bayesian optimization. And what that is in this Bayesian optimization approach, we use a global optimization algorithm that interpolates response surfaces by evaluating only a small subset of all the possible combination. And so we have a search space, which is the reaction to optimize, and so that's our objective function. And then we have a spherical statistical model of the data, which is a Gaussian process, and so that incorporates the uncertainty, and that's how we're modeling the search space. And then with an acquisition function, we can decide where in the search space do we want to look next, to determine what's the optimal sample or experiments to run. So we're balancing between exploiting new areas and then exploring areas that exploiting areas that we know and exploring new areas. And we generate that data, we put it back into the model, and it provides us another set of conditions. And to get to that Bayesian optimization, some 4,500 reactions were run as a training data set so that we could select the Bayesian optimizer parameters. The heat maps here show the yield, reaction yield for experiments in each of the data set. And here's an example of that optimizer at work. It's a Mitsunobu reaction. The standard conditions are in red. The standard conditions give you about 60% yield. But you can see on the graph on the right that with each successive set of experiments that we run, we're approaching much higher yields. And so really by the fourth iteration, we have 3 hits in which we get very high yield for this reaction. So that sort of speeds up our ability to hone in on what the right reaction conditions are in this process invention space. Now, the tool that we created was in-house created with Princeton, so it's a little clunky to use, and we've been looking at packages out there, and I think we're honed in on one particular one, synthetics, that really have taken the same idea and put it into a really friendly user interface for us to be able to really expand this out to more of the organization to be able to do this. Okay, well, sometimes it turns out we can't do this sequentially, is that the route invention is very much tied into process invention. How do we figure out the intermediates? And it's very much tied into the unit operation. And the example I have is actually not from Bristol Myers Squibb, but from Eli Lilly. And this is tirzepatide, who also known as Mounjaro or Zepbound. But this is the obesity drug that they can't keep on the shelves, that they just can't make enough of it. And one of the reasons why is it's a very complex molecule. It's a 39 amino acid peptide. 2 of the amino acid are not natural, at number 3 and number 20. And then it has a side chain on here. So this is a very difficult molecule to make, and it's 4,800 molecular weight. The dosage is really low. If you assume it's 10 milligrams per week, you only need half a gram to be able to treat a patient. But they're estimating they may need to treat 70 million people, and so you need 54 kilos when you look at that number. Now, 54,000 kilos is actually not that big, but it is very large for this particular synthesis. So how do you make peptide synthetically? So one way to do it is solid phase peptide synthesis, developed some 60 years ago by Bruce Merrifield. Essentially, you anchor your first amino acid onto a solid phase, a resin. You activate the next amino acid, and then you couple it, and you repeat this cycle until you've built that chain, and then you can cleave it, and you have the peptide. The advantage of doing this in solid phase synthesis is you don't have to purify in between. You can automate it, and there's a lot of commercial equipment enabling you to do that. The problem is if there's an error that occurs anywhere in the sequence of what you're building, you're going to now generate a lot of impurities because of that one error, because you can't remove it until the cycle is complete. And you actually have to use a fair amount of excess reagents to drive the reaction in this type of synthesis. And so it's not very sustainable. So another way you could do it is through liquid phase peptide synthesis, where people have come up with soluble anchors, PEG, ionic liquids, hydrophobic polymers. The disadvantage is that you have to have a separation step between every coupling that you do. So you either have to precipitate it, extract it, do some sort of filtration. The advantage is you get a higher purity, you're using much less reagents, but then your separation time adds to the cycle time, and you can't really automate it. So you can see how the decision on what unit operations you're going to do really determines how you think about building this particular molecule. And what the Lilly team did was they realized they could not build this molecule using just solid phase or liquid phase peptide synthesis. So they decided to use a hybrid approach where they're going to use both solid and liquid phase. And so they broke it up into 4 distinct parts of the peptide. And they had to make a decision. Do they do 2 parts or two fragments or 3 fragments and 4 fragments, and thinking about the overall cycle time, because now for each fragment you're now going to then couple it in liquid phase. And so there's a whole connection of thinking about how you break up the molecules with the unit operations. But they settle on these 4. And so what they're doing is using solid phase peptide synthesis to create each of these 4 fragments. And then bringing these fragments into a liquid phase peptide synthesis, where after coupling two of the fragments, they would then run through the separation scheme. And they did this continuous. And the nanofiltration is an innovation that I don't think people have been thinking about, that they can apply it to organic solvents after they've been peptide. But this was an easy way for them to remove all the impurities. And so once they form the next one, they could then couple until they get to the final molecule. And this is what their process scheme looks like. And they filed a patent for this entire process, is that they'll take the two peptides and run it through do a continuous reaction. They have an online HPLC that allows them to monitor how well it's going, so they can adjust any feed rates appropriately so they can minimize impurities. And once they've made the material, it goes into a surge vessel, which then feeds into nanofiltration. And they continue this, then take the material and get it to do the next liquid phase step, eventually getting to the final molecule. And so they built a facility in Ireland, and it actually just recently won the Facility of the Year award from ISPE. Now, the story isn't done, because once they have the final molecule, they actually have to do two chromatography steps, which is a very unproductive unit operations for small molecules. And so they do a primary purification, and this is a HPLC chromatography they're doing. But what they did was, when they thought about the fragments to build upon, they were thinking about how much burden it would have on this particular chromatography, again, to think about the cycle time. So this is a really good example in thinking about both the route invention and the process invention. Okay, so now we have a process, and it's time to generate a data set that the FDA is going to want to see. And this brings into quality by design that the FDA had put out some 30 years ago, which is the drug substance has some critical quality attribute, and you have to determine what that is. Typically, it's the purity or impurity profile, the material property. And your process is going to have some set of process parameters, some of which is pretty critical, that will determine whether you're going to meet your critical quality attributes. And setting the specifications on both the parameters and your attributes is a key part of what we call the control strategy to be able to make that molecule. And by saying you have that control strategy, you're telling the FDA that we think we can make this material of the same product quality and the same process robustness based on what we've designated. And the data that feeds into that control strategy really comes from all the work that you've done, whether you've done some one factor at a time, your screening data, your DOE, your high throughput experimentation in your lab, your pilot plant, and your plant runs has generated data about that process. And so the thought is, we should be able to use that data to help us come up with some predictive models about its robustness. And what do we mean when we say robustness? And so what it means to us is that we can meet routine manufacturing expectation. We can run this process anywhere in our network with no expert oversight and achieve quality outcomes. Because in development, we have a lot of oversight over what is happening, but when we send it out to our manufacturing, that same oversight doesn't exist anymore. So you could take a deterministic approach, which is what we've traditionally done, is say, well, as long as the process parameters, as long as the process parameters are within a design space, we're going to meet that critical quality attribute. But as I mentioned, we've already generated data throughout the process development. Why not use that data to predict what that robustness is? And so there's a probabilistic framework first proposed by John Peterson of GSK in 2008 to take a Bayesian approach to thinking about the design space. And that is saying, for the process parameter, as long as it's within this particular space, the probability we meet the quality attribute is greater than a reliability. And so we think this approach enables us to quantify risk and uncertainty using this probabilistic analysis, because we can now incorporate inherent and process parameter variability into any model. And it significantly can give us more information about some of the risk as well as design space than a typical mean response model. And so what is this approach? We use a Bayesian framework where we generate a mathematical random number generator to represent the quality attribute. So that is a probability distribution of that parameter, of that quality attribute, given the process parameter, your model parameters, and any stochastic process parameters. And the way it works is, if you think about a process that you have, if you run it the same 20 times, you're still going to get a distribution of results. You're not going to get the same 20 results, even though you think you've run it the same time. So the processes have an underlying true distribution for a response. So we can estimate these distribution given the knowledge and data about the process. And then these model distribution can be used to quantify the variability. So how does that look? We've got some data set. And so let's create a model to estimate that distribution. But then incorporate the Bayesian aspect of regressing the model parameters, because each of the parameters has some probability distribution. And then we can add in what we know about our typical input process parameter. These are the noise variables. You know, when you set the temperature to 25 degrees, it's not going to be 25 degrees the whole time. It's going to be a distribution around 25 degrees. And so when you put all that in, you can then come up with a probability distribution of your parameter and understand how likely is it going to meet or fail your specification. And you could propagate that even further and think about, okay, if we change the different parameters, what does that design space look like in terms of what combination of parameters allow us to meet our particular specification? And so I have a really nice example from some work that was done at BMS for this particular compound. And so this is the final step to make this particular drug substance. And it turns out it forms this impurity, which is the identical compound, except it's an atrope isomer. And so reaction occurs, you do a distillation to change the solvent, and you typically generate about 4 to 5% of this impurity. And then we go into a crystallization where it's an anti-solvent, cool age. But because the form is different, we can't seed. So there's a nucleation event that happens to lead into the crystallization. So you may get some of the impurity incorporated into your desired molecule. So we do a heat cycle in age, and our goal is to get it down to 0.8%. And we thought, well, if we know that it's getting incorporated, is there an advantage to having what we call a wet mill, a shear device to break up the particles and then do the heat cycle? And our initial data shows that it looks like it's slightly better. But anytime you ask manufacturing to add a unit operation that's not traditional, they're very resistant to that. So we needed to have a really strong data set to say, this is really advantageous for you to do it. And we use this approach to make that case. And so we created 3 probabilistic model, one of the reaction and distillation, and one of the crystallization with and without the wet mill. And here's the results of that, is that the means are pretty similar, but you can see the probability of failing is much higher if you don't have the wet mill. And you can also see that your failure rate goes down very quickly with a shorter age time in that step after the milling. And so there's an advantage to use the wet mill. And so we can convey this, that your cycle time will be lower and you're going to have a lower probability of failing a batch. So that would be advantageous. And that was successful in getting the manufacturing colleagues to say, okay, we'll put in a wet mill. So this is a really great example of using this to forecast robustness, because it allows us to communicate the risk. It gave us a really much more rigorous determination of the design space. And it gave us insight in really a very non-obvious improvement to de-risk the process. And we could use this further to lay out what are the different conditions if you change a particular parameter, so that you understand when you are likely to fail. So this is looking at 4 different parameters, each column, each row, and then each of the X and Y axes of the different parameters that are important in that process. Okay. Okay, so now I've talked about the 3 stages we think about. It's a very intentional design across all the unit operation. The complexity of the chemistry requires a high knowledge generation, and the statistical and probabilistic approach allows us to minimize some of the experimental burden in developing these processes. How am I doing on time? Good, okay. So I want to talk about innovation and high throughput experimentation and data-rich experimentation is really something that is continued to occur. So that's throughout the work that we do. Want to point out the work from Connor Coley and Klaus Jensen in thinking about how do we enable machine learning for synthesis, the data creation work, the open reaction database, but also assessing new reactivity in terms of other types of chemistry that we can be doing to make these complex molecules. And so these are some of the activities going on. And I'm going to talk a little bit about photochemistry. So photochemistry is not new. It's been around for a while. There are chemicals that are made industrially with some photochemical step, but it is new for pharma. And one of the reasons why it's now more advantageous to think about it is the technology advancement in LED and also the development of a lot of new photocatalysts that can transfer the electrons that's being initiated by having the photons. And what photochemistry enables us to do is give us access to really highly saturated or 3D motif that really is very difficult to build otherwise. So there's new bond forming methodology that expands the synthetic toolbox. And it is also a much more green way to form the bond. The disadvantage is it's very difficult to scale up in batch due to the light penetration, and the reaction kinetics is dependent on the reactor geometry as well as the light source. And so the way we think about the photochemical reaction rate is the quantum yield, which is the amount of molecules that you can transform based on the amount of photons absorbed. So that's typically less than 1. So that's dependent on reaction conditions and wavelength. And you then need to multiply the rate of photoabsorption, which is based on a number of things, the molar extension coefficient, the conversion, concentration, your medium, how much light that absorbs, the radiance of your light source, the reactor geometry. So it's a very challenging scale-up problem, but we do know when we scale up, we want to do this in flow because photo flux decays exponentially with the path length, and there's a reason why nature has figured out that to get photosynthesis, you want a lot of surface area. And so flow allows us to maximize the surface area to volume ratio needed for efficient use of the reactor volume and decreasing the residence time. So in thinking about optimizing and scale up photochemistry, there's a lot of parameters to consider. So in addition to your typical reaction parameters, you now have to think about your radiation related parameters as well as your reactor related parameters. And most of these parameters are coupled. So it's a really tricky scale up problem. And it's made even trickier because the type of equipment that we use as we increase in scale actually changes. So it's not like using a similar type of equipment for each stage of development. And what we've done is think about this in terms of how do we decouple some of this information so that we can predict scale up, regardless of how we generated the data to be able to understand that particular reaction. And so we can do high throughput experiments to determine what the reaction parameters are, and that should be the same. And we can characterize the photophysical property of both the reaction species as well as the optical properties of the reactor. And with that information, you can then think about that we can now predict how to scale up in flow. And so quantifying light, we use spectroscopy, spectrometers, measure what the absorption is for the light source as well as for the reactive and product species. And then we can look at the kinetics through our high throughput experimentation and convert that to be able to, that gives us the rate of the reaction. We have all the light source information, and that gives us this quantum yield, which is the missing parameter that we need for thinking about scale up. And so we take that apparent quantum yield and then put in the conditions in which you're going to do that flow scale up. We can now predict what that looks like in terms of both resonance times and conversion. So this is that same chemistry, which we now use the model that was generated to both predict, and the points are the experimental points. So these are areas that we work on because we know we're going to have photochemistry steps coming up. And so we want to be prepared to have that capability when it comes into a synthesis. Okay, collaboration. One of the, a critical component of innovation is to drive collaboration at the pre-competitive stage. And so when we think about it, you know, the IP for pharmaceutical company are in the molecules. It is not in how we develop it. So we want tools that enables us to speed that development. And so about 7 years ago, pharmaceutical companies formed this enabling technology consortium to think about how we can collaborate, develop tools and technology to be able to accelerate the work that we do. And so it's an opportunity to work across companies in industry to talk about what are the problems that we have. And these are the areas that we're most interested in: crystallization, analytical, drying, high throughput experimentation, computational data science, solubility, and one of the more recent one, novel chemistry reaction. And so it's a model in which it allows companies to collaborate and pull their funds together to move projects forward that we're interested in. It's also an opportunity for us to share knowledge of what we think are the problems in the different unit operations. And so a number of publications have come from this Enabling Technology Consortium telling the broader technology community, here's what we think we need in crystallization. Here's what we think we need when we think about drying. And the advantage for the companies is that we can pull funding to create these projects, because each company wants to spend some money, but they can't fund every project. And so by pooling it on a project that we're interested in, we're able to do it. The other advantage is that we have created a templated agreement with ETC. So the technology vendor just needs to sign an agreement with ETC, and they can work with a lot of different companies, as opposed to having an agreement with this company and then agreement with another company. And so these are some of the projects that have come to fruition. A screening tool with microfluidics with Manish Singh's group at UIC, Mettler Toledo creating an online UPLC, creating a modeling cloud for a pharmaceutical company to use some of these specialized tools. In terms of the Novel Chemistry Reaction Environment Working Group, they've put out a paper about thinking about what the reaction technology is needed in some of this new chemistry, electrochemistry, photochemistry, and biocatalysis, and actually funded a project to try and figure out how might we scale up organic electrochemistry at University of Wisconsin with Shannon Stahl and Marcel Schreier. So they're evaluating different types of electrochemical reactors using very typical pharmaceutical compounds. And so that's an aspect of thinking about collaboration to drive innovation in the pharmaceutical industry. Okay, so I've talked about the type of molecules we're making, the need for innovation, the role of collaboration to drive innovation. Certainly a critical element to support innovation is our people. And to get the best ideas of the people, we have to focus on these elements. And so it's how do we develop people, what's the culture in which they work, and how do we engage the next generation? And I think the key challenges are effective technical leadership, creating that inclusive culture and fostering diversity. And so one of the things that we do here at MIT and in our industry is that we hire people for their technical excellence, their ability to do science. We look at where they go to graduate school, what accomplishments they have when they're here at the job. We look at what they've contributed to the project, how many papers they might have published. And when they do that well, we say, that's great. Go be a manager and leader. And now you have to drive the science, which is a different skill set. When you're driving the science, it's about mentorship, motivating, inspiring others. It's about how do you build an effective team, develop people? How do you remove barriers for people to do the work? And so I think this is a really bad model. So people agree? I don't know. My personal opinion is a bad one. So I think there's a step that we have missed, and that's what I call leadership preparation, which is teaching people how to set the expectation, talking to people about emotional intelligence. How do you understand how different people communicate, and really having a strong awareness of your strength and weakness, and understanding what leadership and culture means. And so this is something I have been working on and running my group for the past 15 years. And I wanted to share it more broadly. So so this is the course that I've taught at UVA, is a course to actually think about leadership in a technical field. And so this is how I've organized it in terms of the module, the 4 modules. And so I had a blast teaching this course, and it's something that I'll continue to advocate. So creating an inclusive culture really drives innovation. So it's about creating a culture that really fosters knowledge sharing and seeking, so that people want to share that knowledge, creating a safe place to challenge the status quo, and removing barriers so that everyone can contribute. And so we think about it from an organizational perspective, and how do you put in place that really rewards people who share knowledge, not just create knowledge, but that they're sharing knowledge, having that cross-functional project teams, the matrix management. And when you think of technology, it's thinking about you need both expert support, and how do you going to democratize these new tools that you have, because you need everyone to be able to participate, not just have some people who are specialists. And it's also thinking about flexibility and ways of working, whether you're on site or remote, and how do you manage these plant and lab assignments? Because we recognize everyone's juggling a lot of balls in the air, and you don't want any of them to drop. And then fostering diversity and inclusion. I think companies have done a really good job in thinking about this in terms of creating people and business resource groups, so you have a sense of belonging with some community that's outside of your particular work group. And I think fostering diversity is also creating allies through dialogue. And I think everyone knows about this. Does everyone know about this film? Who's seen this film? I ask this question every seminar. Okay, so this is the highest number, 5. Okay, if you're in science, you have to see this film. This is really important. And it's about these 6 women, and this is Sylvia Sayer, who was one of the female instructors that I had. And it's about engaging the next generation. So at BMS, we have been doing a program that brings in undergraduate women in chemical engineering to learn about what we do, but have them interact only with other women chemical engineers. So maybe not for the undergraduates here at BMS, but for a lot of the undergrads that come, they're astounded that there are so many women chemical engineers. They haven't seen that many in their programs. And we do a lot of polling and surveying to see what impact it has. And it really helps them understand what chemical engineers are and have a better idea of what they might want to do and what education level they want to have. Do they want to go on for a PhD? So we talk about all these topics with them. And then finally, I think we all have a role in engaging the next generation, because we need more engineers. So Take Your Child to Work Day is a really big event for BMS that occurs on 3rd Thursday in April annually. And as Chris had shown, I was really, this 5-minute talk was probably my most important talk that I've given, because I've talked to 213 middle school girls about staying in math so they can be engineers. And so that pretty much concludes my talk, in which You know, I've given you a sense of the molecules, the innovation, how we do development, how we think about collaboration, and how we think about developing people, and just a recognition of some of the folks who did the work, my former colleagues at BMS.
Questions?
So very great talk, and I have a question. Is that you mentioned the data sharing or like public data for machine learning project?
Yes.
So I wonder, like, for the industry, there are many, like, for the pharmaceutical companies, there are many, like, high quality data set, and in the seminar launch, you say that when these companies like share the data, they would do some pre-process. And in this case, it actually it decreases the quality of data set. So I wonder, is there any, from the industry point or interview point, like what is the best way, like for those for the data sharing that they can contribute to the development of this research field, but also protect their IP?
Yeah. So BMS has contributed reactions to data to that open reaction database. We have to look at it carefully to make sure, because as I mentioned, our IP is in our molecules, and the structure of the molecule is important IP for us. So, you know, most of our compounds we work on do not make it to manufacturing. And so there's a lot of data that we can release over time once we realize that this program is no longer something that's an IP issue. So we are continually to reviewing that data set and trying to provide it. We have to get it clear through legal, but I think not just BMS, but other companies have contributed to that open reaction database. We think that is important to contribute to. There's a lag time for us to be able to do that.
Yeah, excellent talk. I really enjoyed it. So you were mentioning these new technologies like photochemistry, electrochemistry, flow. I think often these technologies can actually give you a new route to a compound.
Yes.
So maybe you can tell us a little bit how BMS is implementing them, because I think sometimes these new technologies can then change completely the route of manufacturing, right?
Yes.
And yeah, maybe you can share a little bit.
Yeah, so that occurs in the route invention. Is if there is, we're looking at it. See, you know, is photochemistry something that can do a transformation that really is advantageous, that it eliminates 3 different transformations that if we didn't have photochemistry to do. So it's constantly being evaluated, and electrochemistry as well. But it has to be a significant advantage over what we can do with organic chemistry as well right now for us to then take the next step, because it does require us to think about new manufacturing technology that in some cases doesn't exist, particularly in electrochemistry. I don't know if that answers your question. So it's a constant evaluation, and once there's a clear advantage, we will figure out how might we try to scale it up.
Yeah, but if you look at the timelines, like, is it then actually like then getting implemented?
Well, the development timeline is several years, right? So we're doing route invention fairly early, and so there is hopefully time to figure out how might we be able to scale it up. But that's why the pre-competitive collaboration in trying to think about how might we scale up electrochemistry, how might we address photochemistry is going on now, even though there may not be an active project. So we do need to be prepared for the potential of having that as a step in a synthesis.
Other questions?
Jane, it was a great talk. You talked a lot about how to think about accelerating this process from idea into the manufacturing floor and how to build a culture around that. It was really, really fantastic. My question is really on the backside of that, maybe asking about speculation. Where do you see the innovations and changes needed in the manufacturing to make some of these small molecules more accessible and ultimately overcome some of the shortages and other things that we're seeing now in the marketplace?
Yeah. There's a number of things that are playing into the shortages. Some of, a lot of the manufacturing does not occur in the US anymore for pharma. It occurs in India, it occurs in China. And there's an aspect that the FDA isn't able to keep up with, ensuring all these manufacturing are meeting all the quality standards. So some of the shortages are because some of these facilities, aren't producing material of sufficient quality. So I think there's a lot of folks who want to see, how can we bring back some of that manufacturing back to the Western side? And, the Eli Lilly example is that they're building 2 new manufacturing places. It's in the US and in Europe. So, that is one aspect that the companies have to commit to, bringing that manufacturing back into the US. And manufacturing organizations are a clunky organization because, they want to, be able to make the product, but, they're not driven. There's no advantage for them to put in new technology. It costs them time and energy. And so it really has to come from the development saying, this is a real advantage to do this. So has to continue to evolve. And I think there's a role for the FDA to put in incentives to make it easy to put in new technology. Because right now it's pretty difficult. You put in new technology and the FDA says, well, we don't really understand this, so we're going to put all these barriers for you to, make sure you meet all this. So, there's an aspect of, what the FDA is doing that's really important as well.
I think those are both awesome. I mean, I totally hear that second one, and it feels like small molecules is the place where we should be able to overcome those relationships faster.
Yes.
We think a lot about
that's right. Yes, faster than that, because we have the capability to measure what we are making very accurately. So we could say, okay, we put this new technology in, but clearly here's the impurity profile. It is the same. Yeah. Oh.
I'm getting my steps in, so this is good.
Dan, don't give me a tough question.
Hi, I'm Daniel Patience. I don't think it's tough. I appreciate your experience and your talk. It was really awesome how you overlaid the whole development life cycle and what you have to do to get a drug to market, or a process at least. And your experience comes mostly from big pharma. So what would you say to companies and small pharma who still have to achieve all those same goals and quality and such, but sometimes they don't have, we can't always do the fancy modeling and such. So what would we prioritize? What would you say to people if they're going to work in small pharma companies that might be 100 people? What should they prioritize on?
Yeah, well, a lot of the small pharma is probably more focused on the discovery, right? So they don't have the people on the back end as much to be able to do all this. And they rely heavily on contract manufacturing out there. I will also say a lot of the small pharma are waiting to be bought up by big pharma as well to get their payday, right? So they may not be looking beyond that. I would think, the high throughput experimentation is probably one of the game changers for you to be able to generate data. So if you don't have the expertise to do that modeling, but a lot of the modeling packages are now really becoming more accessible. So again, that could help them decrease the experimental burden, but the combination of those two are really powerful.
It was an amazing talk. Thank you so much for being here. My question, I was wondering if you have any advice for women from the new generation that want to make an impact in the field and that's mainly, dominated by men.
Well, it doesn't have to be dominated by men, is my argument, is that you should continue and that you absolutely belong in doing this type of work. And so I would say continue to persevere, be resilient, and there are a lot of opportunities. My group at BMS is about 35% women. So hopefully, the more we have people in this in this field, the less male dominated it will become.
I don't see any other hands. So thank you, Jean, for talking to us, not just about what sorts of new technologies and approaches are needed, but how they should be achieved through collaboration, through pre-competitive commons, and through actually cultivating a workforce and a culture where everyone can be successful. On behalf of the Department of Chemical Engineering, I think I have to go this side to reach you. Thank you very much, and congratulations for being recognized as the Warren K. Lewis Lecturer for this year.
It's been an honor. Okay. I just want to mention that Dan Patience is the current chair of the Enabling Technology Consortium. So if you want to ask him about ETC, he's right here. Thank you, everyone.
Just have